after Jim Stroll, now we have Bill Faloon. This is pretty exciting. Let me tell you a little bit about him in case you don't know. Bill has dedicated his life to identifying medicine and technology aimed at enabling people to live longer in good health with no upper longevity limit. He's an author of several books and articles in Life Extension magazine. He's made hundreds of appearances in media, Phil Donahue Show, Joan Rivers, Popular Science, many other places. He's the co-founder of Life Extension Group and Age Reversal Network. Please welcome Bill Faloon.
Welcome to our Super Longevity Summit. And for those who are not aware, the science of in vivo youth restoration is rapidly transitioning from mice to monkeys and into clinical reality. Clinical reality means people gaining access to therapies to delay and reverse biological aging. To bring the group up to date with what we've been doing over the last 9 years at RAADfest, I created a 90-second PowerPoint presentation with some slides you've seen before, some slides you've not seen, but there's some newcomers here, and they should know what we've been doing over the last 9 years, because what we've been predicting is transforming into biological reality. The prospect of human age reversal is now transforming into how soon can we get it into humans to keep people from needlessly dying. So I hope you appreciate the next 90 seconds, and then I'm going to bring you up to date with technologies that I could not have imagined would be available as soon as 2024. That's just the beginning. I can't wait to describe what's going to be happening. When I left RAADfest, September 11th, at the newsstand at the LAX airport, National Geographic, the entire issue dedicated to the science of longevity, how to live longer and age better. And they told us pretty much everything you've learned through alternative health publications, through attending RAADfest, all the good things about diet and exercise and certain interventions. But the idea is we're making it into the mainstream. And then a couple months ago, the Journal of the American Medical Association, this is the conventional bastion of thinking, they did a huge study with people, and it turned out that those who practice the healthier lifestyles, they can increase their chances of living to age 100 by as much as 61%. And it means that what you are doing, I think all of you are engaged in something to delay aging. You're radically improving your longevity prospects. But that's not why you came to RAADfest. You already know about, I think, healthy eating, maintaining a healthy body weight, etc. What happened is studies came out just a couple months ago. Caused me to have to continue to rearrange these slide presentations because the data is so impressive. Now you look at those two mice. The one on the left is a normal aged mouse, which we don't want to be. It's got sarcopenia, osteoporosis, hair loss. It looks old. It's all shriveled up like an old person. The mouse to the right, it was given an interleukin-11 inhibiting therapy, and it did not age at the same rate. It lived 25% longer, and it continued looking younger. And you're going to see that throughout this presentation, that when we delay aging biologically, keep these animals alive longer, they also look younger on a longer basis. It's a fantastic situation. So interleukin-11, this is a toxic cytokine that we want to repress in our body. And this study, published in Nature, made some headlines. It's involved, this IL-11 is involved in inflammation and fibrosis, and interleukin-11 increases as we age. It literally kills us. And what this study found is that if you inhibit interleukin-11, you extend lifespan in male and female mice, and it's effective both in young and older animals. And why is that so important? Most of us in the room are no longer young anymore, meaning we need to identify drugs that we can put into our body to delay aging and extend our remaining lifespan. And what was most impressive about this study, when it terminated, 40 to 50% of the mice given the interleukin-11 inhibitor, they were still alive, 40 to 50%, only 10% of the control group. So this is a drug we want to put into our body. And this video shows the normal aged mice, just like normal aged people, shriveled up, not as active, their health is not very good, and they die sooner. It's really that simple. And then when you alter the gene, in this case, and there's a couple of ways to reduce interleukin-11, but they altered the gene at birth, and they were able to enable these animals to live 25% longer in better health with a more youthful characteristic. So what does IL-11 inhibition accomplish? Well, a lot. It's why we take rapamycin, why we engage in calorie restriction, intermittent fasting. We want to inhibit excess mTORC1. This kills us. This creates diseases in our body. We're doing a lot to inhibit it. Inhibiting IL-11 does that. It also increases AMPK activity. One of the reasons we take metformin and certain nutrients that boost AMPK activity, these are good factors. Reduce markers of senescence. Wow, the accumulation of senescent cells in our aged body, we want to get rid of those. Inhibiting this cytokine can make that happen. Liver fibrosis. This occurs in people with non-alcoholic fatty liver disease, killing a lot of people. This one drug may save lives from people who are dying from fatty liver disease, many of which could be here tonight, but they're not because it overcome them. Upregulated brown adipose tissue, and when you do that, you induce fat mass loss, reduce fat, reduce lipids, and improved insulin sensitivity. Those 7 benefits by simply inhibiting the interleukin-11 cytokine. So there's a couple ways of doing it. One of the scientists used a gene therapy, and you're going to hear a lot about gene therapy this weekend. They knocked out the gene that produces IL-11 from birth. They extended median lifespan by about 25%. And that's important for a couple reasons. Some people might think, well, do we need this IL-11? Turns out there are redundant mechanisms to do what it's supposed to do in a body, and we can probably knock it out completely and not miss it. At the same time, extend our lifespan. And that other method, by the way, is an anti-IL-11 antibody that we take as a drug to suppress the IL-11 and therefore improve our median lifespan, in this case 23%. This is in older animals given an IL-11 inhibitor, in this case an IL-11 antibody, to knock it out, even with very, very old mice. Now, fibrosis is a term that most people are not totally familiar with, but it's actually involved in about 45% of the deaths that occur in industrialized countries. You've probably heard about pulmonary fibrosis, there's liver fibrosis, cardiac fibrosis. It's a connective tissue disorder in which our cells are damaged and we simply die sooner. Knocking out IL-11 is a way to stop fibrosis and maybe put it in reverse. The tremendous news is this is in phase 1 FDA trials right now, or it's already passed them and it may already be in phase 2. And because fibrosis is such a common cause of death, there's a good chance FDA will approve this drug relatively soon. 3 different pharmaceutical companies putting big money into getting this drug approved to treat people who are suffering from various fibrotic illnesses, but we're going to potentially use it, like we do metformin and rapamycin, off-label to extend our lifespan. And what can we do right now to suppress IL-11? Good news, almost everyone in this room is already doing it. Lutein to help protect our vision, resveratrol for its various anti-aging properties, a new one being it helps inhibit IL-11, and curcumin and beta blockers blockers. That's a class of drugs that can help slow aging, reduce cancer risk. So these are ways to at least suppress IL-11, but they won't be as potent as the drugs. When the drugs come out, we'll let you know about it, tell you where you can get them, and hopefully they'll be affordable so we can use them to live 23, 25% longer in better health. And again, showing these mice side by side. The left, you can see the IL-11 inhibited mice. They're doing better, and they look younger than the control group there. Not given IL-11 inhibition. They look older, they behave older, and they die sooner. We're trying to postpone all this. The Economist, one of the most prestigious publications in the world, September 30th, 2023, that's 3 weeks after last year's RAADfest, they dedicated an entire issue to the search for living forever. And they had one article after another telling us pretty much what you know already about delaying aging, but they let the rest of the world know. Some of the higher end people on this planet reading The Economist were realizing we can live a lot longer if we behave differently. And then the Bloomberg Business Week, and again, a very high-end publication read by some of the wealthiest people in the world, people who may donate some money to accelerate research. They talked about the prospect of living forever, this immortality-obsessed frontier that we are all engaged in. It's making it into the mainstream media. Now, we've talked about George Church since 2016, his work with CRISPR-Cas9 gene editing therapy. Back in 2016, a lot of people were critical. Can you really do this and make something work? Well, FDA approved the first CRISPR-Cas9 therapy December 2023. Everything I'm showing you is after last year's RAADfest. Then a month later, FDA cleared another CRISPR therapy to treat thalassemia. This is an anemia that a lot of people suffer, and this can now be genetically treated in a way that the person will maintain normal hemoglobin. This is a major breakthrough, and Wall Street Journal, recognizing all this, talking about doctors can now edit genes inside your body. And again, you're going to hear a lot of people like Liz Parrish and others talking about gene therapy and how that can be used even today to reverse many, many aspects of biological aging. New England Journal of Medicine, well, this is the Dean of Harvard talking about the new era of gene editing in medicine. When we started in 2016 talking about gene editing, it was still kind of science fiction. It's now moved into clinical reality. People are going to start to be treated with gene therapies to treat age-related illnesses. The Saudis, they're trying to spend $1 billion a year on research, but identifying scientists with the credentials, the competence, the skill to do some of these research projects hard. But they are spending money right now, which is great news. They've got a lot to spend. If they see a project that needs money, they often will write a check and fund that sort of thing. And of course, the commercial companies raising, in this case, $300 million to figure out better ways to reprogram old cells back into young cells and enable people to potentially rejuvenate any damaged organ, potentially enabling us to live for a very long time, if not forever. Now, we're going to talk a lot about epigenetic age here for my presentation, and many other scientists will talk about it. And this is a way to identify, looking at DNA methylation patterns, what the risk of age-related disease is, and frankly, how long you're going to live. So the epigenetic age, it measures DNA methylation. It can tell you your biological age, and if you're going to engage in an intervention, some experimental therapy, well, you want to get a baseline epigenetic age test, and then after you're done with the therapy, we'll follow up and see how much younger it may have made you. So this is considered the most accurate way of assessing whether people are aging faster or slower, and we sure don't want to be aging faster. And the good news is there are 2 different companies in Rad City offering this test this weekend. You can have your blood drawn and have this test done and find out how old you are from a biological standpoint. So longer lived people, well, they have a younger epigenetic age. And if you have an older epigenetic age, it may mean you need to more aggressively deal with your overall health, because older epigenetic age, it's associated with lower levels of physical and cognitive functionality, and it reduces your longevity. So this is a very important test, and the good news is companies are offering it. So what happened in early 2024? Well, exosome-rich young plasma, and this was published finally in old rats, it cut their epigenetic age in half. That research is now advancing to dogs. The inside story I'm hearing is it's working as well as in dogs as it did in rats. A study we helped fund, young plasma reversed epigenetic aging in old rats, increased their median lifespan, the human equivalent of about 7 years. That's with young plasma. And then OSK, Yamanaka transcription factors, reversed epigenetic age, improved overall health, extended remaining lifespan. And we're evolving that research, our research group, into monkeys, as are some other research groups, which is good news. And we're going to try to make those old monkeys grow younger, and we want to replicate everything in rat mice models when we can, move into monkeys when we can, and you're going to hear some people talking about potential clinical research being done with OSK right now being done. You're going to hear about that at RAADfest. So I'm going to explore that exosome-rich plasma study because it's really important to understand. This was given every other day for 8 days, and it cut old age in half. This is an unprecedented finding published February 2024. And what's impressive about this is the dosing regimen is practical. It was given every other day for 8 days. And who wouldn't want to go into a doctor's office every other day for 8 days, have exosome-rich young plasma put in your body, and cut your epigenetic age in half? I don't think anyone in this room would not do that. And the idea is make it affordable so everyone can make sure they benefit from it. But to explore this study more, they were able to reverse the epigenetic age in the liver, 74%. And I think of so many people who have died of liver failure, this therapy could save their life. In blood, 64%. And that hints they were potentially rejuvenating the hematopoietic stem cell niche in bone marrow. And if you do that, well, you've got better immunity, you deal with anemia, start producing more platelets, red blood cells, and the heart, a 46% reduction in epigenetic age. That could save people from congestive heart failure. There's millions of them in the United States. Hundreds of thousands die. This one therapy could eliminate heart failure as a cause of death. In the brain, about a 24% reversal of epigenetic age. Now, these are unprecedented findings, and these charts, I'm going to make sure everyone in this room clearly understands what they mean. The red is the control group. They're aging normally. They have a high epigenetic age. They're older. The blue that you see there are the control animals, the control young rats, and they have a young epigenetic age. But that orange is what should impress you. These were old rats given the young plasma, and in the blood, their epigenetic age returned almost to that of a younger animal. And the same was true with the liver. The reversal of epigenetic age back down to that of a younger animal. These are life-saving therapies. In the heart, reversal of the epigenetic age of the heart back down to that of a younger animal. And again, the red are the control group. They're aging normally. We don't want to do that. The blue are the young animals. We wish we could just stay young. But with this therapy, we can potentially put this into our bodies, and we are going to be experimenting with this therapy on monkeys to see if we can't reverse aging in monkeys the way it was done in rats. And again, the brain, about a 24% reduction in epigenetic age. It's the hypothalamus. We want to do way better than that, and I'm going to show you some slides on how we're going to do that. Another measure of epigenetic age, the tissue of the old rats. Again, that red is where we don't want to be, but it happens to be where most of us are, and i.e. we're aging, and it's reflected in our epigenetic age score. The blue, the control rats, the young ones, wow, if we just stay young, it'd be fantastic. But with the exosome-rich young plasma, the old rats start to resemble that of the younger animals. That's in the blood, that shows it in the liver, shows it in the heart, shows it in the brain again. Two different ways of measuring epigenetic age, both showing aging going in reverse. But what about conventional markers of health, like liver function? We get older, and you see those red dots? Those are old rats whose liver function is worsening. This is something that happens to people, especially if they're obese, non-alcoholic fatty liver disease, or alcoholic fatty liver disease. Either one, you want to do something about it. And then the younger rat, it's got healthier liver function. But look what happens when you introduce exosome-rich young plasma into old rats. Their liver function on all these 4 tests, look at that, it is improved. It goes back down to that of a younger animal. Older animals given exosome-rich plasma have their liver function markers restored to that of a younger animal. And conventional cardiovascular risks, triglycerides, they go up as we get older, and we don't really want that to happen. The young rats have lower triglycerides, but when you put that young plasma into the old rats, their triglyceride levels go down. And in fact, what you're going to continue to see here is if we can use young plasma and some of these gene therapies, you won't have to swallow so many pills. Your aging process is going to reverse in a normal way, and you're going to stay healthy. HDL beneficially increases in response to the young plasma. Cholesterol decreases. And what's most exciting, glucose. So many of us have glucose control issues. And I say that in the context, ideally, you want your glucose level, your fasting glucose level, to be under 85 milligrams per deciliter. That's ideal. It's very challenging as we get older. We lose insulin sensitivity, and we try to target a little bit below 100. And some of us can't even do that, even with anti-diabetic medications. In this case, exosome-rich plasma moved that glucose level back down to that of a younger animal. And then kidney function. So many people suffering chronic kidney disease. Creatinine levels going up in the old rats, just like it does in so many older people. We don't want that to happen. The young rat, of course, has lower creatinine levels, so its kidney function is better. But look at that. The old rats given the young plasma, their creatinine levels goes back down to that of a younger animal. This could be a potential treatment for people with chronic kidney disease, so they don't have to go on dialysis, don't have to go on a waiting list for a transplant. It's absolutely miraculous. These three measures of kidney function all improved in response to young plasma. And I guess as a caveat, I should let people know, I don't sell young plasma, just so you understand. I'm letting you know what the research findings are. We're going to study these in primates to see if we can't accomplish the same types of results as was done in these older rats. So to put it succinctly, young plasma, old rats, saw their epigenetic age in blood, liver, heart reduced to that comparable with younger rats. This is a major, major advance. And the exosomes, for those who are not familiar with them, they're secreted by our 37 trillion cells, and they enable these cells to communicate. Just imagine if we didn't have exosomes being secreted all the time to have our body work in a systemically effective way. Well, we wouldn't survive. So exosomes, they're secreted by hematopoietic stem cells to boost our immune function, our blood volumes, and then mesenchymal stem cells, and anti-inflammatory properties, and tissue repair. We like healthy exosomes in our body, and that's what our mission is with the old monkeys. Put lots of healthy exosomes into their body to enable them to live longer. Now, this study was published this year, and what it enabled us to do in concert with interacting with the scientists themselves is identify how many exosomes does a 150-pound person need to emulate the rat study, where aging, biological epigenetic age went in reverse. Well, we need about 4.8 trillion exosomes in each treatment. Every other day, young plasma plus 4.8 trillion exosomes, and that would emulate the rat study. But before people do this, we suggest let us do it in primates. Let's see how well it works in older primates to see if we can reverse that. But to put this into context, some people are undergoing umbilical cord plasma infusions. And they're good, except they only contain a few 100 million exosomes. And this study showed that 4.8 trillion, a lot more, produced those incredible, incredible results. For those who want to read more about this, you can go to our Age Reversal website. You can read these studies and to see if you have a neurodegenerative condition. There's a lot of evidence out there that young plasma might help. And anyone considering that, please undergo some blood tests first and undergo the treatment and do a follow-up. See if what's being published in literature applies to you. The more evidence we're able to gather, the more confident we'll be in recommending people do that. I'm going to take a water break and play a little commercial for you that I created. It was a little bit different than the original.
Today we celebrate the first glorious anniversary of the information purification directives. We have created, for the first time in all history, a garden of pure ideology, where each worker may bloom, secure from the pests of a contradictory thoughts. Our unification of thoughts is more powerful a weapon than any fleet or army on earth.
fun making that, and I think you know what side we're on there. So this is another young plasma study that our group helped to fund. We do fund millions of dollars a year in biomedical research. Almost none of it has anything to do with making money. We want to figure out how we can live longer. In this case, we simply used young rat plasma, put this into older rats, and we get some really interesting findings. This is the charitable group that we fund that does this research. We have a number of charities that we support to do research that has no commercial applicability but could result in advancing the science. So what we did here, beginning at the human equivalent of about 60 to 65 years, every other week, we had the rats injected with young plasma, not exosome-rich plasma, just young plasma. The control group didn't get any treatment. And the survival curve over a period of 26 to 30 months, well, it declined, of course, in the rats to not giving the treatment. If you're over 60 to 65, people start dying off. But the treatment group, the young plasma treatment group, none of them died over that period of 26 to 30 months. None of the young plasma animals died. Now, this is median lifespan. We're not talking about maximum lifespan, but we at least showed something that if you're programmed right now to start developing some fatal diseases, the young plasma might delay that. And this chart demonstrates that. The gray dots on the left are the control group, and their median lifespan is reduced. And you can see them dying. Every time you see them dropping, that means more and more of them are dying off. The young plasma group in red, they're living longer. They're not dying at the same rate as the control group. And then on the right, you see some photos. And you see a control rat there looking older, and the treated rat remaining looking younger. We're seeing this consistently with the research. We don't expect it. We're just looking to see how long can we make these animals live, and how healthy are they, but they also look younger. So the good news is when we start to reverse aging, you might start to look younger, and I think a lot of people want that. But the DNA methylation in these older rats in response to young plasma every other week, it improved, and we were able to see improved insulin sensitivity, better immune function, and reduced inflammation. Huge problem with aging. We have chronic inflammatory reactions that destroy our brain, our kidneys, and we just start to age to death. But here's the interesting caveat from this study we funded. Remember, we extended median lifespan 9%, the human equivalent of about 7 years. But the longest lived rat in this study was in the control group. And that rat happened to have very good genes. It let it live longer. It didn't even need the young plasma. It just continued to live. And what does that tell us? Well, we need to reprogram our genes so that we emulate the longest lived people in the world, the longest lived animals. And if we do this in such a way, and I'm going to describe it in just a minute, using OSK Yamanaka factors, we could theoretically reprogram our old cells back into younger cells and have them regenerate for an indefinite period of time. Transcription factors are proteins that help turn on and off specific genes. And if you put in the right transcription factors, you can reprogram old cells back into stem cells or younger cells to continually regenerate tissues throughout our body. So we're going to talk about now Yamanaka transcription factors. These were discovered in 2006, and there are 4 of them: OCT4, SOX2, KLF4, and c-MYC. The acronym is OSKM. You're going to see that acronym used throughout presentations this weekend. You're also going to see it in the popular media because the media is starting to pick up on the fact that this transcription factor combination is able to restore youthful functionality to old cells. And what we're doing with our research is we're only using 3 out of 4 of these Yamanaka factors, the OCT4, SOX2, KLF4, and we're seeing better results with 3 out of 4 as opposed to using all of them. So I'm going to try to depict a little bit about how we're achieving the delivery of OSK into the cells. That's very, very important. So in this case, all 4 of them, and many studies have used all 4 of them being delivered into old cells. And one option is to have your old cells removed and then have them reprogrammed with those Yamanaka factors so they convert back into a pluripotent stem cell, and then that can differentiate into healthy young cells to help regenerate your body systemically. But that's a burdensome way of accomplishing something. So our newer way of approaching this is we're not going to use the c-MYC anymore. That stimulates, by the way, excess cellular propagation. This is great for an embryo, but for older people, we we may not need that. Just 3 out of 4. The OSK embedded into old cells, and then allow those old cells to partially reprogram back into a younger cell. And then we don't want to just let that go. We want to maintain the youthful cells via intermittent expression of OSK. Deliver that OSK into the cells and turn it on and turn it off so we achieve an optimal amount of cellular rejuvenation. So we get you from an 80-year-old back down to a 30-year-old. And if you're happy with that, we just keep you to the 30-year-old simply by delivering more OSK into your cells as the cells use it up. So what happens when you introduce these transcription factors into old mice? Well, you learned 2 years ago, it reverses aging systemically. The Salk Institute people published this 2022. I talked about it at RAADfest. It made international media headlines. The idea, for the first time, aging systemically reversed. Old mice growing younger again, a major, major breakthrough. And what happens with partial cell reprogramming is, well, it can work in vivo, in whole live mice, and these transcription factors did not produce toxicity, did not increase cancer rates. They did what we intended them to do, and again, I mentioned younger looking skin. This is what we're seeing in response to these rejuvenation therapies. People look younger. Inflammation is decreased, reduced senescent associated secretory phenotypes. We're reducing the senescent cell burden in response to these therapies. Now, those studies were done 2022. Look at the date of this study, 2024, in which aging was again reversed by a group of brilliant scientists that we are interacting with right now. And what they were able to do, cyclic induction of the OSK expression in old animals. These were about 77-year-old human equivalent. And they were able to increase remaining median lifespan 100%. That's huge. Now, that's not the whole life of the animal, it's its remaining lifespan. But that's important for all of us in this room because we're older. If something only worked in young animals but failed to work in older animals, we'd be rather depressed. Good news is this worked. It improved the health of the older animals in relationship to control mice, profound age reversal in the heart and the liver, assessed again by those DNA methylation epigenetic age clocks, and they were even able to show some Petri dish reversal of human skin in the Petri dish. In fact, there is some patents that are going to be granted very soon, not by us, but they're OSK topical creams. They seem to reverse aging of the skin back down to whatever age you want. This study, using just the OSK, it was able to improve the frailty index score in a significant way. And I think we all know as we grow older, we become frail, we become more vulnerable to falls, fractures, and premature death. So the frailty index score improved. This is a validated measure of aging, and OSK was responsible for doing this. And again, the prospect, and I've seen videos of this, and it's shocking, where the OSK is applied to an old man's skin, and only one hand. And after a couple weeks, couple months, that OSK skin treatment shows very much younger skin. So there's a potential that we get to live longer and look younger again. So the challenge is identifying the best way of delivering these Yamanaka factors inside of cells. Now, the way that's typically done is using a viral vector. And this technology, I hope you're as impressed with it as I am. You're talking about a virus which is 150 times smaller than a typical human cell. It's very tiny, but the science has emerged and has advanced to the point they can embed OSK into a virus and then have the body infected with that virus. This is an innocuous virus, and in doing so, you deliver OSK into the nucleus and into the DNA. So the viral vector is what's proven, and this is what we'll be doing first with the monkeys, but there's also plasmid vectors that people are looking into, and we're very excited about lipid nanoparticles in which we could control precisely how much OSK gets into your body, including your brain. The brain loves to suck up these lipid nanoparticles, and if we attach OSK to them, we could theoretically induce systemic rejuvenation. And then chemical reprogramming. Bear in mind, OSK OSK, these are normal. They're in our embryo. They express themselves when we're younger. They keep our bodies a lot younger, and then we lose them with age. So the idea of chemically reprogramming old cells to start naturally producing OSK may be a different way to induce the expression inside our cells. So we had this remarkable extension in lifespan. And again, these mice were the equivalent of 77-year-old people, and we were able to reprogram their cells, increase their lifespan, improve frailty scores. And as far as the lifespan, control mice, they died after 8.86 weeks, but the ones given the OSK, they lived over 18 weeks. But here's what should shock you more than anything else that I might present here. It turned out, the scientists who did this, by the way, brilliant pioneers, and when you're a pioneer, you don't do everything perfect, but it turned out that they only delivered a fraction of the intended dose of the OSK into the bodies of these older animals. They wanted to deliver a lot more. They only delivered a fraction of it, and they still achieved the remarkable results I just showed you today. And to remind you, the OSK-treated mice living 18 weeks compared to only, and that's like a 10-year benefit for humans. So control mice had a remaining lifespan of about 5 human years equivalent, and then the OSK-treated mice, their remaining lifespan equated to over 10 human years. And if we can double your remaining lifespan, meaning instead of dying in 5 years, you are facing death at 10, well, at that point in time, we may have achieved indefinitely extended healthy lifespans, and then you don't have to die. And again, even if the animals don't live significant longer, they did live longer in better health. So we both extended remaining lifespan, we being the scientific group that we are working with, and they were able to enable them to live longer in better health. So you look at what happened in early 2024, and wow, more happened, by the way, in the last 12 months, and anyone who thinks differently should let me know. More has happened than everything that has occurred in the past. There's never been a conference where this type of information was able to be disseminated, because this kind of information didn't exist back a couple years ago. It's now published, it's now respected, and we are moving this rapidly into old primates. We want to see old monkeys grow young again, and you're going to hear some people this weekend talking about using it in themselves. I'm not advocating that, but there are people who are doing that. So another study that we helped to fund, and this was a rather crude way of introducing these into the body, but we injected OSKM into the hippocampal region of the brain, and what we found was, well, a lot of benefits occurring as far as memory and learning improvements in response to OSKM, literally in a syringe through the skull of the old rats, and we were able to rejuvenate their brains. And the hippocampal region, rather important. We may notice that our memories and our learning abilities aren't the same at our age as they were when we were younger. Well, these improved in a rather dramatic way. So what you're looking there, a 36% reduction in latency time, which is good. It's a good measurement. It takes the old control rat a rather long time to figure out how to get out of a maze. But you treat that old rat with the OSKM, and again, a crude way of treating them, a needle right into the brain, but no, it didn't hurt the animals, by the way. You can see it's much better. It's much better. And this is another way of measuring this, and I don't want to get people's hopes up too high as it relates to the results of the study, because while we improve the learning ability, the red are the control group, the blue are the old OSKM. But the real fortunate ones are the ones that are still young. You can see that they're really doing much, much better. So we haven't achieved total reversal yet, but we're working on it very hard. This, again, is a different graph showing the memory performance. The young mouse is doing really good. The old control are very bad, but the old animal given the OSKM, and that's injected directly into the hippocampal region of the brain, it's improved. It's improved about 30% compared to that old control animal. So the history of OSK and OSKM transcription factors. 2006, Shinya Yamanaka showed that old cells can be reprogrammed back into youthful cells. 2011, old human centenarian cells reprogrammed back into an embryonic stage. 2022, aging partially reversed systemically. Whole body age reversal in a couple independent studies. 2024, we are moving to rejuvenate old monkeys, and as you're going to see on stage this weekend, Old humans are doing some self-experimentation that we're not involved in, but nonetheless, we want to follow what they're doing to see how well it works. And please understand, before 2006, this was all considered impossible. Nobody in the biology field who was conventional ever thought this could be done, and it turns out it was done. Now, I was invited to attend a private seminar. J.P. Morgan Chase, one of the largest wealth management companies in the world, they're losing their customers because they're getting old and dying. So they put on this seminar, brought in some experts from the Buck Institute to teach wealthy old people how to evaluate longevity data, see what kind of lifestyle changes can improve their life, and what they were able to analogize was your body being a depreciating asset. Okay? I mean, getting old, you're depreciating. Maybe put some effort into changing your lifestyle. And what they said was 93% of your lifestyle determined by your choices, your lifestyle choices. That's 93% of your lifespan determined by lifestyle choices, about 7% by the genes you're born with. So think of it, longevity in investing terms. Make the right choices to extend your healthy lifespan. And if you do most of what they say the best lifestyle choices are, they say you can live to be 25 to or 15 to 25 years longer. And I wouldn't agree with that, but they did have a caveat. That includes aggressive screening, including lots of blood tests, tests to look for early stage malignancies and bone deterioration. So preventative medicine, they were advocating, could add 15 to 25 years to your lifespan compared to doing nothing. So I show this at every presentation to remind people that there's a number of ways that you can avoid premature death. That's level 1 of our house of optimal health. If you fail to correct what you see on level 1 and a lot more, you're going to die prematurely. And I've been doing this since 1977, and I've seen people who think I can do part of what I need to do, ignore my blood pressure, ignore my lipids, ignore my glucose, and they think they're okay. Well, they're not. They do die prematurely, and they didn't have to had they paid attention to the importance of level 1. So you're going to be looking at a lot of individualized age reversal interventions this weekend. Please don't forget level 1. These can be picked up with conventional blood tests, conventional markers of your bone structure, body composition, and we have an opportunity to optimize level 1 and then move into many, many experimental age reversal therapies. That's level 2. But if you don't have a foundation, that's at level 1, it's going to crumble, and then you're going to die prematurely. I don't want anyone to do that anymore. I'm getting very tired of it. Now, there's another way to measure biological age that doesn't cost anything, and that is the Levine-Horvath phenotypical age model. And this has undergone some criticism from people, but please understand, this is based on over 9,000 people, analyzed data over 23 years to see how long they lived. And they were able to identify just 9 blood test markers that predict your risk of morbidity and mortality. And these 9 markers, well, they're on a standard CBC chemistry test and CRP. That's the only one you have to add. Standard blood chemistry test, CRP, can measure your biological age in a surrogate way. The DNA methylation test is a more direct way, but this is a way for people to understand how they are aging. And if you have an old phenotypic age, well, you want to do something about that. You want to put those factors in reverse. And we were validated this year. Huge study done in the UK, 148,000 people using their stored blood samples. And it turned out that if you had a high phenotypic age, you doubled your risk of early onset lung cancer, huge increases in common malignancies that are killing people. So this is a way of predicting your risk of developing a malignancy. And if you have a high phenotypic age, mine is about 14 years younger, by the way, than my 70-year-old, soon-to-be 70-year-old body. So I'm doing a lot of the right things, but I continue to work to improve it. But you can do the same thing. Again, this test does not cost anything to calculate, simply CBC chemistry and CRP. And it was further validated in JAMA. This is a spectacular study. It corroborated what we know about a Mediterranean diet. You live longer if you follow a Mediterranean diet. But they also looked at blood tests. And it turned out that you had reduced death risks associated with lower homocysteine and lipids, lower C-reactive protein, lower creatinine, indicating you have healthier kidneys, lower insulin resistance, lower body mass, and higher alanine and citrate. But these are blood markers that most of you have done at least on an annual basis. And if some of them are elevated, like homocysteine, well, do something about it. Put it in reverse with nutrients, whatever you need to do to lower homocysteine, lower triglycerides, lower CRP. Do that so you don't have an increased risk of death. Now, at Rad City, we've got 3 different blood draw stations that I know of right now. LabCorp is there, and they can do, we've put this panel together, a complete blood chemistry, blood count, plus C-reactive protein, and we added A1C, lipids, and fasting insulin for $85. That's at Rad City. If you have some recent blood tests, that's good. Give us those tests, and we'll calculate your phenotypical age for no charge whatsoever. If you haven't had a blood test in a while, or you've been procrastinating, this is the time to do it. You're right here at RAADfest. Rad City opens at some hour in the morning. I'm not sure which, but you can come in fasting, have a blood test done for $85. Commercial labs charge over $700. Now, some of you had great insurance and you don't pay anything for your blood tests. That's fine. I don't care where you get them, but please have blood tests done as frequently as you can. I have my blood tested on average every 3 weeks. And when I see a marker out of balance, I correct it. I'm not going to let that marker kill me. So this is $85 at Rad City. You can do it after Rad City if you want, but please don't procrastinate. If you want to spend more money, we have the age management panel, very, very comprehensive. And again, it costs a lot of money, retail, at a blood test lab. Some people have great insurance, it's free. If you want to do it at RAADfest, $396. This identifies pretty much everything that we need to do to evaluate your overall state of health, prevent you from dying prematurely. And DNA methylation, epigenetic age testing, True Diagnostics, or the Clock Foundation. They're both here at Rad City this weekend. And if you want the DNA methylation test, that costs somewhat more money. It will tell you more precisely what your biological age is. If you're contemplating engaging in experimental therapy, get this done ahead of time. Don't have the therapy and then wonder afterwards, did it work? Get the baseline, do the therapy, do a follow-up, and let us know what your results are. And if you've not had a DEXA scan or a QCT bone density scan, if you've never done that, please, there's DEXA Body right here at Rad City, and you can get a DEXA scan to measure body composition and bone density. If you have osteopenia, osteoporosis, take steps to correct it. Don't let a bone fracture take you out of the picture. So we know exactly what to do to accelerate our aging process. All we have to do is follow one of these dangerous patterns, and we will die sooner. And please recall, I've been doing this since 1977 professionally, and I've had people tell me, I don't need to do certain things because I'm doing everything else right. I hate to say it, you need to do everything here, or you will die prematurely before we can figure out a way to make you biologically younger again. And if you want to take some experimental steps to extend your lifespan, well, activate AMPK. That's why a lot of you are taking metformin, why you engage in intermittent fasting. There's certain nutrients that help activate AMPK. Senolytics restore youthful NAD levels. Autophagy induction, that can be accomplished through intermittent fasting or rapamycin. And then the biologics, those young plasma, exosome-rich young plasma, those therapies are available. You'll hear people talking on stage about young plasma treatment programs. And then on top of that stair, CRISPR and other gene therapies. That is the ultimate way we might achieve indefinitely extended, healthy human lifespans in our lifetime. The gene therapies are advancing, and you're going to hear a lot about it. But our big issue is we've got the money, we've got a full-time staff of people who want to do a multi-interventional clinical trial. And what does that mean? It means metformin, rapamycin, NAD restoration, aggressive senolytics. It means Depronil, it's a forgotten anti-aging drug, 5 milligrams twice a week, great drug. Young plasma, exosome-rich young plasma, gene therapies. We want to do it all, and guess what? The FDA said no. They said, you're not allowed to do that study in this country because we're concerned about side effects. And our response is, these are older people. They're all going to die if we don't do something about it. And we've got thousands of people who are already doing it, but we don't have a central database to let us know, is it working? Is it really safe? FDA still said, no, you can't do that clinical trial. Even though you've got the funding in place, you've got the staff in place to do it, they said no. It's a major problem. Our human age reversal project, I'm not asking for donations anymore at this stage because we can't do the study. I promised every donor that the only way this money will be spent is to do a multi-interventional clinical trial. We want to incorporate every treatment modality that exists to see how much younger we can make older people, see how much longer we can enable older people to survive. So we've got the money, we've got the staff. I'm paying these people to continue working, but they're unfortunately not able to do what we intended to do. So I'm going to take another water break. This was Joan Rivers and me back in 1993.
Forever, or certainly for a long time. My next guest is the Vice President of the Life Extension Foundation. Will you please welcome William Faloon? Right? Again, another philosophy very close to what we just listened to is your philosophy, which is treat yourself well, think right, and you'll extend your life.
Well, that's correct. We believe in biological intervention into the aging process because of the criminal FDA. That is why we can't get information about drugs used in Europe and Japan that slow down aging.
On one of those drugs, it was back in 1993, I was under criminal indictment, by the way, back then, for promoting the use of off-label drugs to slow aging and to treat certain viral conditions, and many of those drugs eventually got approved by the FDA as safe and effective. Millions of Americans suffered, prematurely died. FDA stood in the way. Going all the way back to 1981, we published an anti-aging newsletter. Instead of Life Extension Magazine, Anti-Aging News, little newsletter. We advocated that thymosin should be approved to treat a wide variety of diseases, 1981, and well, it didn't get approved. FDA wanted the company to do a second clinical trial to validate efficacy. Company didn't have the money for a second clinical trial, so it just fell off the radar screen in the United States. It got approved around the world. Most countries approve this drug, but Americans don't have access to it. And well, some good news is people are now realizing that was a terrible mistake. In 2019, 2020, 2022, these papers are all indicating thymosin is effective and safe and can save a lot of lives, and we still can't get it in the United States. And unlike some of the, let's say, rapamycin you may order from India, like I do, it's very inexpensive there. And we've tested it, by the way, same rapamycin you buy in your local pharmacy. We test not only the rapamycin, we test our blood the next day to make sure it's being delivered into our bloodstream. So offshore drugs, for the most part, very safe. This, unfortunately, comes in a lyophilized form that needs to be kept cool. So it's a challenge to import thymosin alpha-1, which we find very frustrating. But what's happened is, you've heard of the checkpoint inhibitors like Keynote, Opdivo. These therapies break down a tumor cell's wall so it becomes more vulnerable to immune attack. But we're older. We all have immune senescence syndrome, and we don't necessarily have the immune system to attack those cancer cells. Thymosin alpha-1 boosts immune function and reduces inflammation at the same time. And we've got people dying of sepsis, all kind of horrible cancer side effects. Thymosin could mitigate those side effects so that Keytruda, Opdivo, et cetera, will work better, including CAR T therapy. 2024, they're looking at, they being the authors of these studies, independent studies, they're looking at the data indicating thymosin may have saved COVID patients. And the FDA's restriction appears unfounded. Turns out thymosin alpha-1 is showing safety and efficacy beyond what we predicted it would do back in the 1981 period. And this is a study on sepsis indicating that thymosin could turn down an inflammatory response while boosting immune function. So here we are, 42 years, tens of millions of Americans who may have needlessly died because the FDA did not approve a therapy that is now being used in countries throughout the world. We need to tear down the barricade of regulation that is killing Americans. Now, you're going to hear Greg Fahey talking this weekend about thymic regeneration using growth hormone, DHEA, and a new one he told me about, which he's going to announce on stage. But that is a great way to accomplish what thymosin alpha-1 does. But when we get older, our thymic tissue, it shrivels in such a way that we may not be able to regenerate it. The access to thymosin alpha-1 could be a treatment that we all use to combat immune senescence. I can't wait till that happens. Now, if we go back to 1903 and you read the New York Times, they said that it was going to take 10 million years to develop an airplane, that it was in fact impossible to do, shouldn't even be considered. Well, a couple months later, they were proven a little bit incorrect there. The airplane takes off, and then once you prove that concept, that you can fly. Once we prove the concept that aging can go in reverse, then you have all types of people rushing in to improve it. So they go from not being able to fly before 1903 in December to landing on the moon. You talk about an exponential enhancement in technology. Think about that. Think about what's going to happen when we make age reversal a reality. And the good news is you're going to hear, I believe, more evidence disseminated, in some cases for the first time ever, about biological age reversal beyond what I've already talked about. And I don't believe there's been a scientific conference in history, in the whole history of the human race, that is going to disseminate as much information, new information, as you're going to hear this weekend. So I contend that we're further ahead. 1903 to 1957. I mean, even after the Wright brothers flew, by the way, no one thought you could ever create flying buses. They thought that was impossible too. But you know what? People put effort into it. They made it happen. We're closer right now to reversing systemic aging in vivo, in people, than what they were in taking a flying glider and turning it into commercial jet aircraft. So the billionaires, they continue to die, and I hate to say it, they probably could stay alive if they knew where to spend their money. This individual worth $31 billion, and he did donate billions of dollars to scientific research. But from what I could read, it advanced all kinds of sciences except aging. So, I mean, if you're getting older, you'd like to think, put some priority into aging research. No one ever told him, I guess. So he's dead. Someone else is enjoying his $31 billion, and he didn't do everything he should have, in my opinion, to stay alive longer. Now, we have published information over the years. It's available on our website about ways that we think you can delay aging, maybe put it in reverse a little bit, using drugs like rapamycin, metformin, NAD infusions, NAD precursors. There's a lot that can be done, and these interventions are being used today, and we need to systemically keep track of everyone. So I hope you're getting baseline blood tests, engaging in an intervention, getting a follow-up blood test. Let us know how it's working for you. It's very important. This entire presentation available at age-reversal.net. Now, we've got about 5,000 people that belong to this group. And there's probably about 50,000 who want to, but they're afraid that if they give their name and email, we're going to bombard them with emails. We don't do that. The biggest complaint is we don't send out enough, because we only send out something that's clinically significant. We're not going to just throw something in your email box for the heck of it. But if you are interested in keeping up to date with the primate research, with human research, please enroll at age-reversal.net and or watch my whole presentation. And I'm happy here to answer questions, but I've only got 2 minutes to do it. But I will be here the entire time to interact with as many of you as I'm able to. And there's some respected people. There's someone that's got their hand up. Well, I want to thank you all, by the way, for your support since 1977, because the reason we can spend millions of dollars on research that we have no financial interest in is, well, you support us. You support us with blood testing, dietary supplement sales. This is what enables us to fund research, and then at the same time, you hopefully benefiting from what you put in your body. That concludes my presentation, and
Fantastic.
okay.
Thank you. Bill Faloon, ladies and gentlemen.
Thank you, everyone. Thank you, everyone.
Yeah. It's amazing to see the passion that he spoke with on the Joan Rivers show in 1990, whatever, and bringing the passion now. Like that's, if you want to know what the spirit of immortality is, that's like the spirit of immortality right there. Yeah. Excellent.